Bismillah ar-Rahman Rahim. This is the latest paper, March 2023, paper 4.2, and I hope this is going to be helpful to you in the June and the November exam because the paper is made by the same set of people, and so it is very similar to that. So you can get the feel of it, what the paper is going to be like. Now, as you can see, this is the third exam since the syllabus change in 2022, and the time is 2 hours, and there is only one section. There are no section B or section C, so there are no essay questions. And this will be a total of 100 marks, and you know the time is 2 hours. Now, let's go on to the first question. Now, the first question is figure 1.1 is drawing of a longitudinal section of a human kidney. LS is longitudinal section. TS would be transverse section. Use the letters A, B, C, and D. What is A? Cortex, B is the pelvis, C is the medulla, and D is the ureter. So use the letters A, B, and C and D in figure 1.1 to complete table 1.1. So I'm not going to do it. I'm going to give you all time so that you can pause it and do the question and then see for yourself if you can handle this paper or not. Please, everybody must try and attempt to do the paper before you actually look at the video. So for each description, list all the letters that are correct. So location of loop of Henle. Where is that? Where is the loop of Henle? Now, if you remember, the loop of Henle is in the part C, which is the medulla. So location of the loop of Henle is C. Then location of the Bowman's capsule. Where is that? That is in A. And then location of the glomerulus is again in A. So location of the Bowman's capsule and the glomerulus both in A, then contains urine at the final concentration. Urine at the final concentration. Now you look at the marks, there are 4 marks. So here you would have to say B plus D. Why B plus D? D is the ureter. But B is also where all the urine has been made. And now it's collecting here. And then this urine is going to flow into the ureter. So it had to be B and D. So you have to write both B plus D. So you have to write B and D, and that got you your 4 out of 4. Now coming to the B part of the question, the volume and water potential of the urine produced by the kidney vary according to the water potential of the blood. This is a result of osmoregulation. Describe the role of aquaporins in osmoregulation. So aquaporins in osmoregulation, 3 marks. Now before you continue with the video, please pause the video and write your points on a piece of paper and see do they tally with my answer or not. So as you know wording these is the most important thing in A-level biology. So aquaporins, channel proteins that allow water through. 1 mark. Increase the permeability of the collecting duct cells to water. Then changing the number of aquaporins. More the aquaporins, more the cell membrane becomes permeable. So changing the number of aquaporins change the cell membrane permeability. ADH allows more water to be absorbed from the filtrate. Why? Because ADH is produced antidiuretic hormone when the blood is concentrated. So more water needs to be absorbed from that which is going to become the final urine. So more water reabsorbed means that the concentrated blood will now become its normal concentration. So if you've sweated a lot, you've lost a lot of water, your blood becomes concentrated. So you want the concentration of the blood to remain the same because tissue fluid is dependent on that. And we don't want the tissue fluid concentration to vary. So you need to go back to the video on this in which I have talked about this in great length. And coming to part C of the question, describe the role of the brain in osmoregulation when the water potential of the blood increases above the set point. Water potential of the blood increases. So what is going to be happening in the brain? Well, it's detected by osmoreceptors, where in the hypothalamus, osmoreceptors send fewer impulses to the posterior pituitary, so less ADH will be released by the posterior pituitary gland. So very direct question, Very easy to get the 3 marks if you know this part of the syllabus. It's a very important part of the syllabus. Pause the video here. Detected by osmoreceptors in the hypothalamus. Osmoreceptors send fewer impulses to posterior pituitary gland. Just writing to pituitary gland is not going to get you any marks. Posterior pituitary is the most important thing. Less ADH released by the posterior pituitary gland. So there were about 5 mark scheme points, and you have to give me any 3 to get your 3 out of 3. Now, coming to question number 2, this is a question which needs to be read twice. Why? Because it's a little complicated, and you need to either skip this question and do it at the end so that you can finish the paper with the questions that you know. This is a little difficult. This is a higher level. If you have not understood this part of the syllabus, you find finding it difficult. Interferon alpha IFN alpha can be produced as a recombinant human protein to treat some types of cancer. The gene IFNA2, there's a gene which codes for IFNA, IFN alpha. One method of producing recombinant IFN alpha uses genetically engineered E. coli bacteria. So E. coli is a bacteria that contain recombinant plasmids. Each recombinant plasmid contains the gene IFNA2, 3 regulatory sequence of the lac operon, promoter operator, and lac-I, a gene for antibiotic resistance AMPR. Each of the sequences for the lac-I gene and AMPR gene contain its own promoter. As a result, these genes are always expressed in E. coli bacteria that contain this recombinant plasmid. Figure 2.1 is a diagram of the recombinant plasmid. The promoter regions of the lac-I gene and the AMPR gene are not shown. We have a promoter, we have an operator, we have IFNA2. What was IFNA2? The gene that codes for IFNA. This is here, it's written. IFNA2 gene. Because plasmids are made of DNA, so must have the gene for it. So the promoter region of the lac-I gene and AMPR are not shown. So and then what do we have? We have AMPR and we have, so the promoter regions have not been shown. So the promoter regions will be somewhere here just before that. So there'll be a promoter region here, and then if we look at here, then it will be somewhere here because promoter comes first, so it's going to be like this. So the promoter for the AMPR here and the promoter for lac-I here. The promoter regions of the lac-I and the AMPR gene are not shown. The start of transcription of the gene IFNA2 by E. coli with the recombinant plasmid shown in figure 2.1 needs to be controlled to obtain an optimum yield of IFN alpha or IFNA. Scientists investigated the effect of 2 inducers of transcription. You see, we want more of the protein because the protein is needed. It said here the protein is needed for what? Produced to treat the human recombinant human protein is needed to treat some types of cancer. So we need the protein. We need the gene to express more transcription. 100 times transcription, 100 protein molecule made. 1000 times, 1000 molecules made. So the start of the transcription of the gene with the recombinant shown and needs to be controlled to obtain an optimum yield. Scientists investigated the effect of 2 inducers of transcription on the production of recombinant IFNA. Lactose inducers. Lactose, which is converted to allolactose in E. coli, and IPTG. So lactose and IPTG, which is a synthetic molecule with a very similar structure to allolactose. IPTG cannot be broken down by E. coli. So we studied the effect of 2 inducers of transcription. Lactose, which is converted to allolactose in the E. coli, and IPTG, which is a synthetic molecule with a very similar structure to allolactose. The scientists grew 3 cultures of E. coli containing the recombinant plasmid in the same growth medium. The growth medium contained glucose, amino acids, essential vitamins, and minerals. The growth medium did not contain lactose. So it contained only glucose, amino acids, and essential vitamins. and minerals, but they did not contain any lactose. After 4 hours, either lactose or IPTG at the same concentration was added to two of the cultures of E. coli. As a control, the third culture of E. coli was grown without adding lactose or IPTG. The concentration of recombinant IFN alpha in the culture was measured at different times over a period of 28 hours. The results are shown in figure 2.2. So culture to which IPTG added, which one is that? This one here is here. So I'm just going to color it so that you can then make your assumptions about it. Then culture to which lactose was added, this is red. This was which one? This was here, right? And then we had control culture. This was here. So control culture was here. So the concentration of IFNA in the cultures was measured at different times. These results are shown in figure 2.2. So now you look at that, and this is the concentration of IFNA at microgram per dm cube. Now let's try to understand the question. The regulatory sequences of the lac operon contained in the recombinant plasmid are involved in the control of transcription of the gene IFNA2. Explain the role of the gene LAC-I in the control of transcription of the IFNA2 gene between 0 and 4 hours. So 0 and 4 hours would be somewhere up to here. Now you can see here between 0 and 4 hours, there is only 20 micrograms per dm cube of IFNA. So there is less of that being produced. Now that means what is happening? It is preventing transcription of IFNA2. So it is still transcription, but it is preventing transcription. So some transcription is taking place, but not a lot. So the RNA polymerase could be binding after the promoter. So it is preventing transcription of IFNA2. Why? Because maybe codes for the repressor protein that binds to the operator. So maybe there's a repressor protein that is binding to the operator. That is why RNA polymerase is unable to continue with transcription, but some of it is being able to be made because 20 micrograms per dm cube is still being made, because here you can see this is 20. So prevents transcription of IFNA2, or you can say RNA polymerase binding after the promoter. So a little bit of transcription still taking place. Codes for the repressor protein that binds to the operator, but then even if it binds to the operator, even then some of it is still being made. It's only 20 micrograms per dm cube, but it is being made. Then coming to part 2, with reference to figure 2.2, describe the changes in the concentration of recombinant IFNA in the culture containing IPTG from when IPTB was added at 4 hours to the end of the experiment at 48 hours. So the concentration of recombinant in the culture containing IPTG. Now if you look at this culture to which IPTG was added, what is described? Describe is no biology, I always tell you that. Concentration of IFN alpha produced peaks, increases and then decreases. You can see that it increases and then it decreases. Peak is at 8 hours, 4 hours after the addition of IPTG. Why? Because we added the IPTG here at 4 hours. Then increases immediately after addition of IPTG. Increase, you can see there is an immediate increase. And then concentration of IFNA decreases more rapidly from 8 hours to 16 hours. And then from 16 hours to 22 hours. And then you could give me a data quote. A data quote means that you're giving me the figures at 4 hours, and then you're giving me the figures at 8 hours, and then you're giving me the figures at 22 hours. So this is a data code. Data code means that you give me the figures, time in hours, and the IFNA in micrograms per dm cube. So how would you word it? The concentration of IFNA produced increases and then decreases. Simple. Peak is at 8 hours, 4 hours after addition of IPTG. Increases immediately after addition of IPTG, but that was added at 4 hours. Concentration of IFNA decreases more rapidly from 8 to 16 than 16 to 22 hours. And then if you gave me a data code, then you got another mark for that. But you can see there is 5 mark scheme points, but only 3 were needed for you to get your 3 out of 3. Part 3 says suggest one reason for the difference between the concentration of recombinant IFNA in the culture at 8 hours in the presence of lactose and the concentration of recombinant IFNA in the culture at 8 hours in the presence of IPTG. As you can see, at 8 hours, the to which lactose was added, that is only 50. To which IPTG was added, it is 285. So the question they've asked you is, suggest one reason for the difference in the concentrations. And you can see there's a lot of difference in it. Generally a difficult question because you see it's only 1 mark, so suggests are always very difficult. So the possibilities, you can come up with a whole lot of things. Time delay as lactose has been enzymatically converted to allolactose. That was something in the question. Lactose is broken down, so has to be continuously taken up by E. coli. IPTG will be at a higher concentration than allolactose. Or maybe we can say IPTG has a higher affinity for the repressor protein than allolactose, so the repressor protein is removed and more IFNA is made. You see, the repressor protein has to be removed, or lactose may be used up by the E. coli. So a lot of possible answers, and if you can come up with something good, if you can't, well, you can just leave it. So lactose may be used up by the E. coli, or you can say IPTG has a higher affinity for repressor protein than allolactose. Then coming to the part 4, suggest one reason for the change in the concentration of recombinant IFNA in the culture containing IPTG from 12 hours to 16 hours. So what has happened from 12 hours to 16 hours with IPTG? So 12 hours to 6 hours, you can see it here, somewhere here, we're going to start here. It's starting to decrease. So what could have happened here? Maybe less glucose, less amino acid, less nutrients, or you could have said IFNA is unstable and starts to break down. So suggest one reason for the change in concentration. Again, suggest difficult part. Less nutrients in growth medium, recombinant IFNA is unstable, breaks down. Now coming to the B part of the question. The gene AMPR in the plasmid shown in figure 2.1 codes for a protein that provides resistance to the antibiotic penicillin, ampicillin. Suggest how AMPR allows genetically engineered cells containing the recombinant plasmid to be identified. The fact that E. coli that have taken up the plasmid will survive in the presence of ampicillin because they have the resistant, they have the code for the protein that provides resistance to ampicillin. So if we grow them on ampicillin, then these E. coli which have taken up the plasmid will grow, they will survive. They will not be destroyed by ampicillin. Ampicillin is an antibiotic, it kills bacteria. The word resistant to ampicillin, this word resistance to the antibiotic ampicillin means that it's not killed. These bacteria are not killed by ampicillin. So the E. coli that have taken up the plasmid will survive in the presence of ampicillin. If the ones which have not taken up the plasmid will actually die. So in order to find out the ones which have the desired plasmid, we have put in this gene for antibiotic resistance in it. Now coming to the last part of this question, seems to be a very long question, doesn't seem to end. Bacteria can evolve antibiotic resistance through natural processes. Outline how bacteria can evolve to become resistant to antibiotics. Very simple. There's going to be a chance mutation, and this is a form of natural selection, and this is of course directional selection because more of them becoming resistant. So if we have 1%, 2%, 3% becoming resistant, then at the end of it 70% will become. So this is directional selection. Antibiotic acts as selection pressure. Bacteria with the mutation that code for antibiotic resistance will survive and reproduce. They will pass on that allele that codes for antibiotic resistance to the daughter cells by either vertical transmission, that is by binary fission, or they can pass on the allele for antibiotic resistance to other bacteria by horizontal transmission. And of course, other points that we could have given was increased chance of resistance if people do not finish the full course of antibiotics, or some antibiotics may act as mutagens. So all of these points, there were lots and lots of points. In the very simple case, natural process gave you the clue. It was a question on natural selection. But even if you didn't write that, you could have easily come up there about 8 mark scheme points, and any 5 of those you could have got your 5 out of 5. So now look at the wordings of it. Random chance of mutation, 1 mark. Natural selection, directional selection, antibiotic acts as selection pressure. Bacteria with mutation survive and reproduce, pass on the allele for antibiotic resistance to daughter cells by binary fission, pass on the allele to other bacteria by horizontal transmission, and increased chance of resistance if people do not finish the full course of antibiotics. Some antibiotics may act as mutagens. So it is these points which are for every question on natural selection. That mutation and then natural selection, and then they pass on the advantageous alleles to their offsprings, and this of course happens over many generations. And of course there is in some cases there's directional selection like in this case, and also in bacteria there is horizontal transmission. So there is vertical transmission as well as horizontal transmission. Now I would like to explain this to you all, how these is directional selection. You see, you can see here that there is the original distribution, which was this, which is being shown to you by this graph. And you can see there's low drug resistance, whether you give figures here 1, 2, 3, 4, 5, 6, or something like this. And then you give them the antibiotic drugs. So this is the arrow for that. And then what happens? Distribution after directional selection. Now look at it, how it has changed. Now they are more in number of the high drug resistance. Similarly, this is another diagram showing you how the selection, this is directional selection example, then this is stabilizing selection, in which it has narrowed down whatever the phenotypic trait is. And here we have again then disruptive selection, in which you have two peaks. Population after the original population was this one, and then now you have two peaks. These are at a advantage, and this the middle phenotype is at a disadvantage. Then another example, direction selection, antibiotic resistance, normal bacteria, bunch of bacteria including a resistant variety. Get bathed in antibiotic, most of the normal bacteria die. The resistant bacteria multiply and become more common. Eventually, the entire infection evolves into a resistant strain. Then evolution by natural selection, another picture showing you the same thing, how it has changed. A group of bacteria, including genetically resistant ones, are exposed to an antibiotic. Most of the normal bacteria die. The genetically resistant bacteria start multiplying. Eventually, the resistant strain replaces the strain affected by the antibiotic. So, and then we come to the point which I mentioned in the paper, was vertical transmission and horizontal transmission. Vertical transmission is this one dividing into two by binary fission and the daughter cells getting the plasmid, and this plasmid contains the resistance gene. So this is the plasmid which contains the resistance gene. Now both the daughter cells have the resistance gene. But in horizontal transmission, of course, this is not the normal process. What happens is that it is just some other bacteria, and it is going to be transferred to, the plasmid is going to be transferred to another bacteria, could be a slightly different type of bacteria. So transformation, transduction, and conjugation. Horizontal transmission, basically you don't have to know these, but you have to know horizontal transmission is between this bacteria and another bacteria. And this plasmid is just going to be transferred. So it's not by binary fission, but it's called horizontal transmission. A little more detail about horizontal gene transfer, conjugation, bacteria to bacteria through direct contact. Then transformation is environment to bacteria. So the plasmid is in the environment and somehow reach the bacteria. and then becomes part of it, the DNA, a portion of the DNA, and becomes part of a plasmid. Then transduction is phage to bacteria. Phage means it's a virus, but it infects bacteria. It is a virus which likes bacteria. So we've done this in genetic engineering. So the phage is going to inject it into a bacteria, and that is a way of horizontal gene transfer. That completes this for the time being, and we'll continue in the next videos the other questions. We've only done two questions in this video, and please watch out for the next 2 or 3 videos, which will complete this March 2023 exam paper 4.2. Thank you very much.